So, today we're going to talk about RNA processing in your chapter 14 of the book, and we are going to talk about exons and introns in genes, structure and function of 5' cap and poly A tail in eukaryotes, splicing machinery, and alternative splicing. Now, I'd like to talk about the collinearity concept. It means that a continuous sequence of DNA transcribes a continuous sequence of mRNA, which encodes a continuous sequence of amino acid in a protein. Most of the prokaryotic genes are collinear. In bacteria, mRNA is directly used for translation, versus in eukaryotes, there is a pre-mRNA that is first transcribed from DNA and then processed to create mature mRNA for purpose of translation. In an mRNA molecule, both in eukaryotes and prokaryotes, each amino acid of the protein being translated from the mRNA is specified by a set of three nucleotides called a codon. In both prokaryotes and eukaryotes, mRNA contains of three primary regions: 5' untranslated region, protein coding region, and 3' untranslated region. In bacteria, transcription and translation occurs simultaneously, as you can see in this figure. In prokaryotic mRNA, there is a consensus sequence of nucleotides at the 5' UTR called Schindel-Garner sequence, which is the ribosome binding site during translation and is located on the upstream of the start codon, shown here in yellow. We are going to talk about the Schindel-Garner sequence in the translation chapter. The protein coding region determines the amino acid sequence in the protein and starts with start codon and ends with stop codon. The 3' UTR affects the stability of the mRNA and helps regulate the translation of the mRNA. Unlike prokaryotes, most eukaryotic genes are not collinear. Scientists performed an experiment which allowed DNA to complementary base pair with mRNA, and the results were that sections of the DNA looped out, as you can see in this figure. Their results showed that the entire DNA sequence is initially transcribed, but many sections are removed from the RNA. Exons are part of the DNA sequence that end up in the final mRNA. Keep that in mind that RNA transcription from the DNA always starts and ends with exon sequences. Introns are part of the DNA sequence that are removed from the pre-mRNA. In eukaryotes, there are also some sequences at the beginning and end of the DNA that are not getting translated: the 5' UTR and 3' UTR. The UTRs do end up in the final mRNA, thus they are part of the exons. Mature mRNAs undergo three processing events: addition of a 5' cap, addition of a poly A tail, and splicing out the introns. In eukaryotes, transcription takes place in the nucleus, whereas translation takes place in the cytoplasm. This separation allows eukaryotic RNA to be extensively modified before translation. In this figure, you can see the location of the 5' cap and the poly A tail. The addition of 5' cap happens rapidly after the initiation of transcription. The cap consists of 7-methylguanosine attached to a pre-mRNA by a unique 5' to 5' bond, which is completely different than the 5' to 3' phosphodiester bond. The process is shown in this figure for your information, so you don't need to know about this in details. The 5' cap functions in the initiation of the translation. It increases the stability of the mRNA and influences the introns removal. A string of about 50 to 250 adenine nucleotides is attached to the 3' end of the RNA right after the cleavage site. Poly A polymerase is the enzyme responsible for adding the adenine nucleotide. Functions of the poly A tail consist of increasing the stability of the mRNA. It plays a role in the export of mRNAs from the nucleus to the cytoplasm and facilitates the attachment of the mRNA to the ribosome. Introns are removed by RNA splicing. RNA splicing takes place in the nucleus before RNA moves to cytoplasm. Splicing requires three sequences in the intron called the splicing code. 5' splice site, also called the splice donor, and 3' splice site, also called the splice acceptor, are the first two sequences we're going to talk about. These splice sites consist of short consensus sequences and begin with a GU at the beginning of the intron and at the 5' splice site, and ends with an AG at the end of the intron at the 3' splice site. These sequences play a crucial role in splicing, and mutation in any of these sequences may prevent splicing. Branch point is an adenine nucleotide over here that is located 18 to 40 nucleotides upstream of the 3' splice site. The branch point location is really hard to determine, and it's almost impossible to locate without doing an experiment. Mutation or deletion of adenine in the branch point may also prevent splicing. Splicing takes place in the spliceosome. Spliceosome is one of the largest and most complex of all molecular structures, and it consists of 5 small nuclear RNA and 300 proteins. snRNAs are coded by genes, and they are functional RNAs that provide the enzymatic function of the spliceosome. RNA splicing consists of 2 steps. In the first step, the pre-mRNA is cut at the 5' splice site that frees exon 1 from the intron. Then the guanine nucleotide in the 5' end of the intron attaches to the adenine nucleotide in the branch point and creates a lariat. In the second step, a cut takes place at the 3' splice site, and the 3' end of the exon 1 covalently attached to the 5' end of the exon 2. The intron gets released as a lariat and gets debranched and degraded in the nucleus. In cystic fibrosis, there is a mutation at the branch site of intron 8 and directly affects the splicing of exon 9. The way it works is that the guanine nucleotide in the 5' end of the intron cannot attach to the adenine nucleotide in the branch point because of the mutation, so it skips intron 8 and finds the adenine in the branch point of intron 9 and splices the pre-mRNA. The problem is that it not only cuts out intron 8 and intron 9, but also exon 9. Nearly all individuals with cystic fibrosis express a small fraction of CFTR mRNAs that lack exon 9 and express a non-functional protein. So, the presence of the introns in an organism increases complexity of that organism. In prokaryotes, we have none to very rare number of introns. In yeast, only 3% of the genes have introns, 200 among 6,300 genes. In fruit fly, most of the genes have 3 to 10 small introns. In human beings, most genes have introns. On average, there are 8.8 exons and 7.8 introns per gene, but the average size of introns is 2000 bases versus 200 bases for exons. So, alternative processing pathways work in a way that a single pre-mRNA can be processed in different ways to produce alternative types of mRNA, resulting in production of different proteins from the same DNA sequence. One type of alternative processing that we talk about today and in the class is alternative splicing. In alternative splicing, the same pre-mRNA can be spliced in more than one way to make different mRNAs that are translated into different amino acid sequences and thus different proteins. In this example that we can see on here, we have 3 exons and 2 introns. The first way we can splice this pre-mRNA is to splice intron 1 and intron 2, and the mRNA contains exon 1 followed by exon 2 followed by exon 3. The other way is to splice out the intron 1, exon 2, and intron 2, so our mRNA contains exon 1 followed by exon 3. In human genome, we have only 21,000 genes, but more than 70,000 different proteins. Around 70% of human genes are alternatively spliced. Over here, we have a review of what we talked in pre-mRNA processing in eukaryotes. We had the addition of the 5' cap that facilitates binding of ribosome to 5' end of the mRNA. Also, it increases mRNA stability. We had the 3' cleavage and addition of poly-A tail. The poly-A tail increases the stability of mRNA and facilitates binding of ribosome to the mRNA. And we had the RNA splicing. RNA splicing removes non-coding introns from pre-mRNA. It facilitates export of mRNA to cytoplasm and allows for multiple protein to be produced through alternative splicing.